And now onto this final class. We might again take a bit longer, and I'm very sorry, but this is the last time I'm doing this. Also, the exercises will be a bit more relaxed this week. There's just 3 exercises for you to practice how the exam could look like, and you can ask any questions to your TAs. So the session might be a bit shorter so that you still have your break. But yes, essentially our last lecture was supposed to be next week, but as I have told you before, next week is a workshop that we are organizing at EPFL, where you are all warmly welcomed. I sent your registration form through the emails maybe 2 weeks ago. The deadline for the registration is passed, but you have a special status as the students of this course, and you can still fill it. It's going to be meaningful for us to know how many chairs we need. So what is the goal of this last lecture of me to you is if you were to attend the workshop now or the conference on artificial life, you might be surprised that there will be a lot of topics that we did not cover in the course. That is because there's a lot of contemporary examples of cellular automata, especially continuous ones, which overlap much more with the field of machine learning that we did not have the time to cover, and where also the theory has not yet really caught up with the existence of the examples. So there is not so much to teach, but I think they're very interesting. So you can see this last part as a way to get you up to speed, such that if you would go to an A-Life conference, you would be able to tell from most titles what the talks will be about. So I will just very briefly go over the topics that we did not have time to cover in detail in class, unfortunately. So the first one that we did not get to cover deeply is looking in more detail in the space of cellular automata. And specifically, we did not discuss what are some heuristics with which we could automatically detect interesting looking behavior, and we did not discuss where can we find such behavior in the space of, let's say, 2-dimensional cellular automata with radius 4 and 10 states. And the answer is that it is actually very difficult to do this. But if you were to sample cellular automata from some large space, chances are you would most likely encounter some variants of these 3 behaviors. So some kind of ordered phase where activity quickly dies out, some kind of chaotic phase where you don't see many patterns form, and then the interesting phase where we have some gliders forming and interacting with each other. So the question is, if you're a statistical physicist, the question is, can we introduce some kind of parameterization of the space of cellular automata with some kind of temperature, which would allow us to study some critical values of this temperature where the interesting behavior can happen. And in this context, it would have been interesting to mention the work of Christopher Langton, whom we have already encountered when we discussed the self-replicating Langton's loop. So essentially, Langton did suggest a heuristic both for parameterizing the CA space and for estimating the complexity of a cellular automaton's trajectory, such that from certain numerical experiments, he ended up with a picture like this. So the x-axis is the parameter, which is here called the Langton lambda. The y-axis is some measure of complexity, and the appealing picture that is often mentioned is that around certain critical region, the complexity of the cellular automata explodes. So this is not anything proven. We can view it as a hypothesis, but let's look into slightly more detail on how this was done. So when we have systems at criticality that have certain complex behavior, they have certain classical features. One of them is that the time the system takes to converge to its typical behavior usually explodes around criticality, and this is exactly the kind of way Langton decided to assess complexity here. So essentially, what is typical behavior here? So very briefly, when we have a particular trajectory of a cellular automaton, if it was on a finite grid, it will eventually start looping, but this time could be very long. So let's instead measure some kind of, instead of the actual configurations, let's measure some observations of it. For instance, it could be the density. So we just compute the average number of the states in the whole configuration, and we see how this evolves. And then we can say that the system converges to typical behavior if this density stabilizes around its mean by no more than some number of standard deviations away, for instance. And this is already a good enough approximation to get something meaningful, because both the ordered phase actually converges very fast to its true attractors, let's say. And in the chaotic phase, the actual convergence is very long. But if we look at the average density, this stabilizes also very fast. And then there are certain systems which also contain the interesting looking ones, where we can have more interesting fluctuations. So the average convergence time is longer, and this is essentially what this figure shows. What is the parametrization? So this is a very, very rough parameter, because it is just a property of the local update rule of a cellular automaton. In the simplest case, you can imagine a binary cellular automaton. By NQ, we can denote the number of transitions of the rule table that go to the quiescent state, which could be, for instance, 0. And K is the number of states. Then the lambda is just essentially the ratio of the rule table that does not go to the quiescent state. So if we have just 2 states, if this number is 1/2, it kind of means that the actual rule table is balanced. So exactly half of the inputs go to 0 and exactly half go to 1, which is a necessary condition for surjectivity and for other notions of chaos that are more stronger. So essentially, the chaotic behavior roughly could be kind of contained in the region of lambda being 1/2. And you can also imagine that if lambda is very close to 0, essentially almost all inputs immediately go to 0. So it is very improbable that any activity will sustain in the grid for long. And as you can see, as you tweak this parameter, maybe something interesting will happen. But then again, measuring CA's behavior just from its local rule is a very, very rough estimate of its behavior. Nevertheless, it becomes an interesting way to sample in very large rule spaces, where chances are, if you just did that naively, you would most likely encounter the chaotic looking picture. But you can, for instance, sample the rule tables according to some bias given by lambda. And if you skew that, then you might maybe find interesting behavior with larger probability. But proving, or coming up with the perfect way to measure complexity and with the perfect way to parameterize cellular automata are essentially open problems, and proving any kind of relationship between the two seems extremely difficult. But another topic that would have been interesting to cover is that we can study related families of dynamical systems to cellular automata, where exactly such analytical results and identifying critical regions becomes analytically feasible. So one family of such systems are called the random Boolean networks. What you see here on the left is a picture of a standard 2-dimensional cellular automaton with von Neumann neighborhood, except for now every node represents a cell, and each cell is connected exactly to its neighbors. So this is essentially a connectivity graph given by the neighborhood, and I just highlight that here the local update rule is the same in every cell of the grid. The architecture of a random Boolean network is different and more randomized. and depends on a few parameters. So first of all, we have n, the network size. So it is at every point just a finite graph. So here we have 8 nodes. And for each node, we randomly sample some incoming edges. And this will define the node's neighborhood. So for instance, the node 1 just has one incoming edge from node 3, so it just has one neighbor, which is 3. So when it wants to compute the value of its next state, it will just depend on the value of F3, or like of V3. Whereas, for instance, this node 5 has 3 incoming edges, so 3 neighbors. So given a network size and an average connectivity, which is just the average in degree of a vertex, we can sample random Boolean networks with that particular connectivity. And secondly, we randomly sample a Boolean function for each vertex, which corresponds, or whose arity is exactly the in degree of that vertex, which will essentially be the local update rule for that particular vertex. So we don't even have uniform updates because we use different functions for different nodes. But we can all sample them with the same bias, which is just the probability of that Boolean function outputting a 1. So hopefully this is clear. This network essentially is just a binary one, so each vertex can have the values 0 and 1, and it was originally used by Kaufman who studied them as very simplified toy models of gene regulatory networks, where you can essentially represent or see each node as either expressing or suppressing a gene, and these influencing each other through these local updates. With certain cell types or phenotypes being expressed as a whole, kind of corresponding to the attractor of such a system. And experimentally, Kaufman noticed that if he samples such networks in an unbiased way, so p equals 1/2, there is some critically looking phenomena for the average connectivity being exactly 2, as the network size goes to infinity. This was later proven by Derrida and Pomo using a very simple approach, which is just the annealed approximation. So what they were able to do is define this critical line, which is the exact relationship between the average connectivity and the local update bias, such that in some sense, if the connectivity is too low, then the system kind of is in some ordered phase, and if the connectivity is too high and there's too much activity going on because of it, we are in some chaotic phase, and then you have this critical line, which is in the critical phase. Very roughly, the way they do it is they look at the randomly sampled initial configurations and a small perturbation of that configuration, and they measure how the distance between these two evolves over time, where at each time they resample a potentially different random Boolean network with the same parameters to compute the next time step, and they look at this in the thermodynamic limit, essentially obtaining the fact that if the relationship between k and p is like this, the distance after a few time steps essentially decays exponentially fast, and in this region the distance explodes exponentially fast, and here essentially it decays but much slower, like in a polynomial way, for instance. So what we can then do is to randomize the architecture of cellular automata in more sophisticated ways. So here you see a different architecture yet, which is now closer to cellular automata. This is again a finite network, which is now a random deregular graph. So here every node has the same neighborhood, in this case 4, and we apply the same local update rule in each cell. And we can still obtain certain analytical results about phase transitions in such systems, but using much more sophisticated methods, which is, for instance, what the group here at EPFL did. But we don't have the time to go into too much detail. Yet another way to introduce randomness into cellular automata is to keep the connectivity network the same, but randomize the local rule. For instance, instead of having a classical rule table, we can introduce some parameter p, which is the probability of the output being 1, and then we can study systems like this. Just to mention one very interesting result by Nassim Fates is that if we have the following problem, if we study binary cellular automata that classify whether there was a majority of zeros and ones in the initial configuration by just converging to either all zeros or all ones. If we only limit ourselves to one-dimensional binary deterministic cellular automata, it has been proven that no such CA can successfully compute this problem. But if we randomize them, we can solve this problem with arbitrary precision. So in some sense, the randomness can even aid with certain types of computations. And more about noisy cellular automata and how we can design them to be robust to certain noise perturbations will be given by the workshop at the time of the actual lecture, which is next Thursday at 2 p.m. by Ethan Lake, who was working on this with Ehsan, who's from Berkeley. Yes? Was there a question?
Yeah, I was just wondering, when we consider an automata with probabilistic outputs like this, do we understand it as it being resolved after each iteration? Okay, so like at each iteration is like a new rule table that determines...
It's more, so the rule table stays like this, but the way you generate space-time diagrams follows the distribution where when, yeah. Mm-hmm.
Sorry, I think it's the same question, but I don't understand. Does it mean that if at one precise step, for instance, 0, 1, 0 gives 1 in some cell, does it also have to be the same on the other side?
No. So every time you encounter, so if you have some configuration and you want to update it, every time you encounter a configuration of the local neighborhood, you sample. So, and it's not the same then for all the configurations you have seen like that. I'm sure there's also variants of how you can, what kind of probabilistic cellular automata use, but I think this is the most classical one. Moving on to a whole other family, which I think really dominates in the interest of the general artificial life audience, is continuous cellular automata. So if we first made some quick comparison, from what we have seen in class, for instance, in the context of self-replication, discrete cellular automata are quite great for engineering if we have some desired behavior we would like the system to have. For instance, to be locally universal or to have some self-replicators, because we have the freedom to really design the local rule and the components in some compartmentalized ways. But it is much harder to, let's say, discover new unexpected behavior, because we would have to have a good sense of what is interesting behavior, to be able to detect it in some automated way, and to develop some optimization methods in the discrete space that would optimize for such behavior. And unfortunately, we don't have much control over the discrete space of cellular automata, which is vast, and doing one small change to a CA's rule table can make a huge qualitative change of its behavior. This is where continuous cellular automata come in. These are, in general, more difficult to engineer if you want to see some desired behavior, but they are much better suited for discoveries of new behaviors. Now, their landscapes are much smoother, so they're much more amenable for some natural parameterization that we can then sweep through. And even more importantly, they're amenable for continuous optimization methods, such as gradient descent methods, so that we can search automatically for certain behaviors. So we will have a quick overview of that. But first of all, what do I mean by continuous cellular automata? So there's different combinations of what can happen. There's essentially three aspects we can look at. There is the space, which in the discrete case is just integer lattice. the states, which is a finite set, and time, which is just discrete updates. And the continuous analogs of this for us will essentially transform Z to R, transform the states to either the real line or some interval of the real line, or some powers of the interval of the real line. And yeah, the time would turn it into some differential equation. We don't require a continuous cellular automaton to be continuous with all these aspects. For instance, we can still have a discrete grid which has where each cell has a continuous state. So one of the most iconic examples of a continuous cellular automata is called Lenya, and in the grand scheme of things was not introduced so long ago, specifically in 2018 or 19 by Bert Chan. So what are we working with? Lenya lives in 2 dimensions. So technically, the space here is really R², and the states are now, can take any real value from the interval from 0 to 1 including. But technically, we will still have discrete time steps, but we will talk about how this can be quite nuanced of a debate. So suppose we start with some discrete cellular automaton which has some, for instance, radially symmetric neighborhood. We can interpret the neighborhood either as a subset of Z², which is finite, or as its characteristic function, which is a finite support, right? Now, in the continuous case, this naturally turns into a smooth neighborhood, which we will now call kernel. And essentially, this is again a function from R² to 0 and 1 to the whole interval. So now we can assign to each location not just the binary value, which is whether it's present or absent from the neighborhood, but some weight. And we require this function to also have finite support. In the case of Lenya, to further restrict the space, which would be vast, we want the function k to be radially symmetric. So essentially what it means is that if we want to compute k at some position x, y, it is the same as using a slightly different function just with a single input r, where r is just the distance from the origin. And typically for k, for this function, we use some unimodal function such as a Gaussian bump. It is important that the integral over this curve will be 1, because how will we use this? Okay, before I show you how will we use this, there is a second ingredient that will fully determine the dynamics of a Lenya system, which is the growth function. So this is now also a unimodal function, which not, as opposed to the kernel function, which had values positive which had positive values. The growth function has both negative and positive values and need not have an integral of 1. So how do we put all of this together? In the classical discrete case, for a binary cellular automaton, the configuration is a function from Z² to 0, 1. In the continuous case, at some time t, it is a function from R² to 0 and 1, not necessarily with a finite support. And if we want to update this configuration by the time step delta t, how will this look like? We will look at, at some position x. So here x is a vector from R². You look at the value of the array at this particular position, and you do a small increment scaled by the size of delta t. And how do you compute the increment? You do a convolution of the original array with the kernel at position x. So this is a standard definition of convolution. I'm sure you have seen somewhere. This is where it's important that the kernel is normalized such that this convolution will also have values between 0 and 1. And essentially, this tells us how much is the neighborhood of the particular location x alive. So if the value is very high, it means that let's say, the values that are at the neighborhood of a cell were generally quite high. So somehow this neighborhood is very populated. So this is where, once we put this into the growth function, it would kind of kick in a similar logic as larger than life rules would. So if the neighborhood is overpopulated, actually the growth function gives you a negative value. And the same is the case if the neighborhood is underpopulated. And now if you hit with the neighborhood values a specific sweet spot, is where your increment is positive. Now if you did this, you might be wondering, how do we have a guarantee that the output value will be between 0 and 1? And the answer is we don't. This is why we have to introduce the clipping function, which essentially clips all negative values to 0 and all values larger than 1 to 1. And this is essentially the only obstacle that prevents us from taking the continuous time limit. So if we did not have the clip, what we could do is subtract from at plus delta t atx, divide by delta t, take the limit with delta t going to 0, which a team in Japan did figure out how to do. It's essentially a trick. So the growth function here essentially tells us the direction at which we could step. And instead, you can introduce some target function which tells you what should be the target value of that cell. And this helps you to get rid of the clipping function somehow. We don't have to go into too much detail. To then also consider a variant of linear which is continuous in time, which is called asymptotic linear, which I just wanted to show you for completeness. So how do we implement this? If you would like to see a linear simulation of on your computer, we have to backtrack our steps. So we have to discretize the space back to some kind of grid. We have to discretize the kernel accordingly, and we will have to use some floating point values that only have finite precision. So essentially what you're doing is when you're seeing linear on your computer, for instance here, is you're seeing a discrete cellular automaton with just a huge amount of states and a very large grid. So you might be wondering, why do we call these continuous? And there is a big qualitative difference between linear and a very large discrete cellular automaton. That is that for linear, we have this Platonic ideal, which is the continuous system, and we can see what happens at larger and larger precision, both in space and time. We don't have such a prescription of how to refine the system for a discrete cellular automaton. So here, for instance, we have the radius, which is a parameter essentially just telling you what is the size of the kernel. And here we have the number of time steps per unit time. So we can, for instance, increase this, which essentially decreases delta t. You can see the system still, this beautiful soliton that we had survived, but now it's just very slow to update this system. So let's go back. What we can also do is increase the spatial precision, essentially by increasing the kernel size. The way you can see it is, if you want to increase spatial precision, you can either have a larger and larger grid, or you can increase the kernel size and then, but let's say keep its absolute size such that more and more cells fit within the kernel in some sense. We don't have a guarantee that the solitons we see in linear will survive this, but what is interesting that sometimes they do. Here, essentially, the creature is just still adapting to me increasing the spatial resolution, which is pretty fascinating. I did double check this with Bert yesterday. So it really is the creature adapting to me increasing the resolution, learning to survive. This is not the case for all creatures. Some of them kind of die when we require this adaptation, but we can, for instance, stop the system, increase the spatial resolution, which also rescales the creature automatically through the way it is implemented, and then when you run it, the creature kind of still exists in this more resolved space. I really recommend playing with this app that was designed by Bert-chan, who manually swept through many different parameters of linear systems and found these beautiful solitons with this smooth behavior. Probably the most classical one is Orbium, that I don't know if I will find. See, I did not play with this. Ah, yes, this is the iconic soliton of Lenya. But you can see, and he essentially went through the effort of basically introducing some kind of artificial biology taxonomy of them. Now, if you explore these different families and genii and whatnot, you see that they actually, unfortunately, do not coexist in the same Lenya system. Essentially, one has to have different parameters to observe different types of solitons. But now we can ask, okay, since we now have the natural parameterization, can we understand the space of Lenya systems? And this is what's happening here. This is just some simple numerical experiments I did. So here I just fix a particular kernel. Once I do that, the only freedom I have is over the growth function, which is naturally parameterized just by the mu and sigma, which is the mean and the standard deviation of the Gaussian bump, which is exactly the x and y axis. And what I, for instance, noticed is that even if you look at different kernels, there is this, let's say, similar qualitative regions of Lenya. So here is the region that no matter how you initialize the system, everything always dies out. So that's like the ordered phase. In contrast to that is this, let's say, expansive metastable region where no matter how you initialize the Lenya system here, the mass quickly expands from a local initialization to the whole grid and then does not converge exactly, but let's say oscillates around these patterns. And then in between these two phases, we have a phase which is actually somewhat sensitive to initial configurations in the sense that if we initialize the system with a small patch of noise, the small patch dies out. And if we initialize the system with a large patch of noise, it goes to the expansive phase. And as a subset of this region, which is somehow sensitive to initial configurations, is the interesting region where some kind of solitons emerge. What is interesting is that often the case, the solitons emerge around this phase transition from the ordered to the metastable phase. But all of this is within the realm of numerical experiments. So this already brings me to the first interesting open question, which is how can we define the boundaries of this region analytically now that we have this natural parameterization of the system? So you can really play with this Explorer too. It's just online. So that is about the classical Lenya. And you can imagine that such beautiful solitons quickly caught a lot of interest in this. And since 2018, we have had a huge number of flavors of different Lenya systems. So the most natural generalization of Lenya is to not only consider a single channel that is values between 0 and 1, but to take some power of that interval, for instance, 0 and 1 to the power of 3, which you can interpret as now 3 values which determine the red, green, and blue values. You will have to have more kernels, for instance, 9, to guide the behavior of such a richer state space. But this leads to colorful Lenya pictures, which are even more beautiful. And it's also interesting to look at visualizations of asymptotic Lenya, which is generally smoother in time. Now, people notice that it really is quite annoying to have this large part of the parameter space where everything either dies out or somehow explodes in activity. And a natural observation is that if we somehow introduced mass conservation in Lenya, those two phases can no longer occur, which could increase the probability of something interesting happening. And that is indeed the case. There's two versions. I will not go into detail in how they differ. One was done by our colleague Vasilis here at EPFL, which results in a richer behavior of solitons. A very famous example comes from the team at INRIA Bordeaux, called the flow Lenya. What you see here is quite fascinating. So what is going on? so in these mass-conserving lineas, very roughly speaking, the update rule does not tell us what is the next state of a cell. It kind of partitions the mass in a cell, and it tells us the directions in which it should flow in the next time step, very roughly. But now we don't need to just flow mass. For instance, here, a part of the state space actually encodes the parameters for the update of Lenya itself. And these parameters can also evolve together with the state. So here the different colors are actually different, let's say, genomes of Lenya, which can now kind of coexist in the same space, meaning that we can have a higher variety of creatures because they can be governed by different dynamical rules in different regions. And what's interesting is they can somehow learn to coexist. So here, this blob essentially means that the inside has this yellow update rule and is somehow encapsulated in a membrane given by a pink update rule. Do not ask me how the coloring of these parameters work, though. To push this idea even further, a team from Google Zurich is also looking at examples of particle systems. So here we have discrete particles flowing in continuous time. Also very fascinating. So to quickly summarize this very brief Lenya chapter, I will give you a few questions that are open that I find interesting. First of all, in Lenya, people seem to focus on these very localized soliton-like structures that move around, often in fascinating ways. But it is not clear to me, despite the fact that they are very beautiful, why are they the only form of interesting behavior? So when we were looking at these phase spaces of Lenya, for instance, we noticed this expansive region of the space, which kind of looks like artificial neurons. So quickly, essentially, these fill the whole space, and then they start interacting through sending each other these signals and excitations. And sometimes they actually synchronize over time to some like more smoother flows of the signals. So in general, yeah, what is the interestingness of a Lenya trajectory? Second of all is an interesting idea of Clément Hongler, which is that maybe you have heard of KdV, which is a particular differential equation governing the, let's say, flow of in shallow waters, where you can have these soliton-like traveling waves, which have a well-defined speed and shape. And there is a very clever transformation of the solutions you can do, which allows you to have a very analytically precise way of telling how many solitons are in a particular picture, even if there are two waves that are currently interacting and they would visually look like one big wave. From that transformation, you can see that how many waves there are. Are there 2 of them or 3 of them? So can we be inspired by this result, though very vaguely probably, to come up with some very clever transforms of Lenya configuration space that would allow us to identify what exactly formally is a soliton. So another interesting thing, which maybe you have thought of, is studying self-replication in Lenya. So there are some very interesting examples of self-replicating solitons, but very rarely can we see ones that would self-replicate, creatures that would self-replicate whose size would go much beyond the kernel size, which in some sense means it is hard-coded in the actual local rule of Lenya. So the question is, can we find self-replicators that are much larger than the kernel size in Lenya? And lastly, what is interesting is that despite the fact that the efficiency of our computers is just increasing exponentially, people still limit themselves to quite small simulations. So the typical size would be like a world or a 2-dimensional grid of size 100 by 100. But what happens if we scale this up? Will it be possible to see qualitatively new behavior in the exact same system just if we give it more space? And this is what people have been experimenting with very recently. So here is a simulation of a much larger flow linear. I think this is 4,000 by 4,000, which in the grand scheme of things is still nothing if we would like to see the emergence of biology or chemistry from this physics of this world. But I think it's a very meaningful thing to look at, and I think eventually we will also be able to zoom in and see how these different parameters coexist and influence each other. So these are the videos generated by my colleague Marco from INRIA Bordeaux a few days ago. So this is quite nice. Probably too dark for us to see properly though, but I can send it to you. So to finish with the last iconic example, in the little time that we don't have, I'm sorry. Our neural cellular automata, which might be worth the explanation though, because these are really, let's say, at the border between artificial life and machine learning. As you can see, the exploding interest in machine learning means that there's an exploding interest in neural cellular automata as well. So a neural cellular automaton lives typically also in 2 dimensions, though not necessarily. But here we will have a discrete space. So we will still think about grids, but now each cell of the grid will have a continuous state space, typically some power of the real line, and some discrete time update, typically just one. So let me again start by recapping or what a discrete cellular automaton does, but in a machine learning language. So if we have a binary grid of cells, well, how does the update look like if we are interested in how this central cell should change its state? So we have this local neighborhood which extracts the values around this state. And then we have some nonlinearity f. That is the update rule which tells us the next state. If we restrict ourselves to a very particular family of these local rules, we can represent them by a particular kernel and some nonlinearity, for instance, sigma. So what the kernel can be is just a matrix which exactly agrees with our neighborhood size. So it's a 3 by 3 matrix, for instance, of binary values, such that we can essentially convolve our grid with this kernel. That is, we overlay it with the neighborhood of the cell and we do pointwise multiplication and add all of these values together to obtain just a single value, in our case a natural number, which we can then feed into the nonlinearity to obtain the value of our central cell in the next time step. This is just a discrete cellular automaton where I represent the local rule in a very particular way, that is by some matrix multiplication with this kernel and then taking that outcome and sending it through some nonlinearity. But if you think about it this way, it becomes extremely natural to now, or maybe you're already thinking the word convolutional recurrent neural networks, where we can essentially just relax all of these to be real numbers and it will be defined just as well, which is exactly what neural cellular automata are. So you can think of the values of this grid to have real numbers. We have some kernels which can also be real valued. We do convolutions to obtain a vector of values, and then we can pass that through some dense layers or through some nonlinearities until we obtain the next value of that cell. So the situation is not that simple when it comes to the exact architecture of neural cellular automata, where we don't just have a single grid or we don't have a single real value. We have a whole stash of them that corresponds to this parameter c, so the number of channels. We can think about it like this. The first channel can represent the value for the red channel, the second for the green channel, the third for the blue channel. The fourth channel somehow determines the activity so that we have a sense whether a particular cell is dead and should not be updated or whether it is alive and deserves an update. And then we have these hidden channels where we allow the neural cellular automaton to learn and represent information as it needs, but we really don't care about this when we visualize the dynamics of the system. A good analogy of this is, in a few weeks I will give you an exam, and there will be the, let's say, exam channel, which is where you write down your answers, and the hidden channels is the extra papers when you can write your auxiliary computations, where you can develop all the knowledge you want, but you don't want me to see because you will only hand the exam in the end. So these are the auxiliary papers that the neural cellular automaton has. So what happens is that now our configuration is this array with a certain height, a certain width, and number of channels. And we have some fixed kernels. In our case, it will be 4. You will see that it's not it's actually not such an arbitrary choice. And what we will do now is, if we are interested in the value of this central cell, we take this slice of the neighborhood of this cell across all the channels, and we convolve, or let's say we apply the first kernel to every single of these channels, which will give us exactly C values. Then we do exactly the same with the second kernel, which gives us the next C values that we just concatenate, and we do that for the third and fourth kernel, so that we just end up with a vector from R to the power of 4C. Now, so what are the kernels? The first one is just identity, so that each cell has a chance to remember what was its state. The next one is the so-called subkernel or filter in the x direction. So you can see it's designed cleverly in such a way that if in your picture you had values that would be increasing from left to right, and you convolved it with this kernel, the value would be very high. If you had a decrease in the values in the x direction, the value of this would be very low. And if there is no change in the x direction, this value would be approximately 0. So it essentially helps the cell detect its spatial derivative in some discrete sense in the x direction. Now the same goes for the y direction. And the last kernel essentially represents a discrete Laplacian, telling the cell how much does it on average differ from its neighbors. So this essentially gives the cell some awareness of its neighborhood. So this is called the perception phase. And now what do we do with this vector of the values we have aggregated? We do the so-called adaptation phase, where we just pass it through some matrix multiplication, we add some bias, we apply some nonlinearity, in this case ReLU, we do another matrix multiplication, and we obtain a vector of the exact dimension we need, which is a vector in R to the C. But this will not determine the actual value of the cell. This will determine, let's say, the step at which we should go. So if we actually want to update our array with some time step delta t, we do this residual update where we take the original array and we add a little step in the direction of delta a t, which is this vector we have obtained. So this is quite a small neural network in the grand scheme of things of what we are used to today. The only parameters that we actually train are these two matrices and this one bias. So these are the parameters theta. So we can now parameterize this function by f theta. And we fix these cleverly chosen kernels exactly like this. So they are not part of the training process, though nothing stops you from training your kernels any way you would like. What we can now do is we can, for instance, choose a particular initial state of this array and some target state. For instance, we would really love to grow a gecko. And we can essentially just train this neural network on a particular loss of our choosing. The most naive way would be just really to take the difference between each pixels and doing the average of the distances, for instance. After some hyperparameter, which is the number of time steps, after which we would like our neural CA to grow from this, for instance, very simple seed into the gecko. And this is exactly, let's say, the first application of neural cellular automata we have seen, which caused a pretty big excitement. This was done by a group of researchers from Google Zurich, together with a biology professor from Tufts University, Michael Levin. So here, so for instance, in the gecko case, it is what you see is one particular neural cellular automaton trained not only to grow the gecko from a particular initial seed, but also for the gecko to maintain its shape. And you can do many more. You can choose different target values, for instance, patterns. So you can have a target value of a particular picture, for instance, containing bubbles, and then you can have a neural cellular automaton that generates this. So how you can think about this is the neural cellular automaton essentially learning representing a very nice compressed version of the pattern that you are generating. So one particular way you could represent the pattern is just through the actual function which assigns to each position the actual value of the pixel. But here the neural cellular automaton is a very compressed way that tells you essentially how to reach that particular picture from some chosen initial trajectory. So hopefully what you could be thinking is, isn't it interesting that the two applications of neural cellular automata is essentially for morphogenesis, that is the symmetry breaking when we want to grow an organism's shape from a very simple initial egg or a seed, and for generating some textures that sometimes can look very much like Turing patterns. And this is not a complete coincidence. On one hand side, you have the work of Ehsan, essentially showing that for certain neural cellular automata, especially the ones that generate textures, you can look at their refinements both spatially and temporally, and you see that the exact same rule works across different resolutions in space and time, kind of showing that what is actually happening is that the neural CA is learning some underlying differential equation, which is kind of invariant to these scales. And on the other hand, you can take actual reaction diffusion systems, even though we did not talk about them in too much detail, unfortunately, either. But here you can believe me that this is some particular form of a reaction diffusion system, and you can discretize it in such a way that it can be represented as an update of a neural cellular automaton, and then you can essentially train these discretized reaction diffusion systems to generate the patterns you want, which was, for instance, again done by the Google team. Yeah, I think this is essentially it. Let's say, an interesting question about neural cellular automata, since we already have examples of neural CAs growing interesting patterns from initial simple seeds or eggs. How can we again use this to have full self-replicators, since we have already, let's say, all we would need is for them to learn how to lay eggs and then they could grow. So why is this not done? And second of all, a question overarching all of the examples we have talked about, which is linear neural cellular automata and reaction diffusion, is what does it mean for continuous systems to compute? This is something we have covered in the discrete case. And it is not any profoundly different actually approach. And there, similarly, how we formalized it for discrete system, there is a paper by Jordan and his co-author Simon Rashikov, who do formalize what it means for a continuous system to compute. Essentially, it just boils down to dealing with Turing machines that operate with actual real values, which don't hide the halting problem, and some kind of notion of robustness, such that in the continuous system you don't have the discrete information represented by very fragile regions. This should be some well-behaved sets, hopefully. But if you're interested in this, then there is a very new result by Jordan and his PhD student about the fact that reaction diffusion systems are locally universal or Turing complete. So this will also be a part of the workshop on Thursday at 4 p.m. if you're interested. And to do a full circle back to the discrete world, just two interesting, let's say, side effects of our continuous, I don't want to call it detour, but our continuous journey of today, is that sometimes if you want to find certain behaviors in the discrete world, you can go through the continuous world and then discretize back, and surprisingly this works. So for instance, you have another, this is sad. Okay. Okay, so let's have a small. So what you see here is essentially a discrete cellular automaton which makes a logo that you might have seen before. It's not a coincidence that this was again done by a team at Google. And the way this works is that the local rule of this cellular automaton is represented by composition of certain logic gates that can be made differentiable. Then they can be trained similarly to neural cellular automata, and then these logic gates can be discretized again in a way that actually preserves the shape that you would like such a discrete cellular automaton to grow, which is quite surprising that some continuous systems survive this brutal discretization. And actually this is the same case for Lenya. This is from an unpublished work, just from my personal correspondence with Bert Chan. So what you see here is the exact same orbium, but discretized to the point where now it has just 13 states and it still preserves its shape. So that's just an interesting remark. And this concludes it. Thank you so much. Yeah, that's it. Are there any questions?